everybody. Let's see. Oh, wow. We have some panelists on there and some participants. I'm waiting for a couple more people to join. Good to see you. All right. Welcome, friends. We're on our second week of getting to know our scientific community. And just a little background about me. My name is Chersty Bourne. I've worked in labs and I've led online trainings, and I even got to impact about 3,000 students when I worked as a STEM and makerspace teacher. I now work for a company called Elim Bio, and today's webinar is sponsored by our company, Elim Bio. Elim Bio is happy to be assisting researchers and scientists on the front lines. Many researchers we are working with are helping find solutions for COVID-19, and we're so proud of all the researchers in our scientific community. Elim Bio knows that students are at home now doing their part to help flatten the curve. And we wanted to take this unusual opportunity to introduce you to some amazing people that are making a difference in our world. There are so many helpers that are helping make a difference in our scientific community and trying to find solutions for positive change. And today I wanted to introduce you to a superhero and a scientist, Dr. Sushmitha Kumar. And Dr. Kumar does her work at Scripps Institute of Oceanography. Her current research focus is biofuels. And this is a really hot topic right now in science, as it could be a great solution to help some people help solve some problems with our energy problem. A little housekeeping before we get started, and I hand over the call to Dr. Kumar. As she's speaking, you'll notice there's a button on the bottom. It says Q&A, and you can type in questions in there, and a box will come up, and it will show that question to me. And we're going to have a couple breaks, and I'll relay those questions to Dr. Kumar, and she can answer them live. So you can even type in when she's talking and I'm talking, and it won't interrupt her talk. Another thing to let you know is that we're going to be putting up a couple polls while we're speaking, and the polls are totally anonymous, which means there's no stress. It's not a test. You can answer right or wrong. We're just trying to get an idea of how much everybody already knows, and that will help us to kind of know which direction to take the call. So I think we're ready for our superhero of the day. Dr. Kumar, welcome. Can you tell me a little bit about you and your current research?
Hi, everybody. I'm so excited for this. Thank you, Mrs. Bourne, for putting this up. This is very exciting. So, okay. So you can see the screen good?
Yes, looks great.
Okay, perfect. So, as Mrs. Bourne explained, I'm Sushmitha Vijaya Kumar. I am currently a postdoc in Scripps Institute of Oceanography in San Diego. So our institution is in La Jolla, but that's where the beach is at. And so I'm a postdoc, meaning that I'm doing independent research. I'm getting trained on doing independent research after getting my doctorate in life science. So today I'm going to be talking a little bit about cyanobacteria and how they can be used to produce biofuels. So basically, can we use microorganisms to drive your cars, to can they replace solar panels, which is something very interesting. So the first thing would be, if you see the slide, I have a question there: why do cyanobacteria get eaten? And that's my overall big question. And you can see these small rod-shaped structures. These are the cyanobacteria, and the blob that's moving around and trying to eat everything is an amoeba. So I don't work on this predator, but I work on another predator, which I'll be talking about at serious. So as we know in nature, everything likes to get eaten up by something else. So which is pretty interesting, and which is what I work on. So I work on predator and prey interactions, and how a predator likes to go and hunt and eat their prey. So Mrs. Bourne, can you put up the first poll, please?
Sure. What is a food chain? There you go, friends. Go ahead. You can answer that question. We're having some people sign in and give their answers. Give a couple more seconds for an answer. What is a food chain? So the options are a long chain made of food. The second option is a process of preparing food. And the last one is a pathway that energy and nutrients flow throughout the ecosystem. And it looks like pretty much everybody answered the last one.
Cool. So you're right. So that is the answer to it. So if you look here, I've put up two food chains. It's like a pyramid. It's a triangle. And you can see that the bottommost is the primary producers. So if you see terrestrial ecosystem, and by terrestrial ecosystem, I mean like land and everywhere we live. So in the primary producers there are basically the trees. So they can like take up the carbon dioxide and sunlight and they grow and give us food. And you know, that can be again consumed by primary consumers. So it could be small animals, insects that can eat them. And then these primary consumers will be eaten up by secondary consumers. So it could be a frog or like a snake or something like that. So what I mean to say is like a food chain is very important. So if you remove the primary producers, the other things that are stacked on top of it cannot survive. So if there is no plants around us, for example, we cannot survive because like we all depend on plants to get our energy from. And you know, if there's no plants, there's no animals, and then we cannot survive. So maintaining this food chain or this food web is very important. So when you look at terrestrial ecosystem like us, you know, plants are important. But when we switch to marine ecosystem, so when you go into the ocean, there are no big plants, but you know, we have something similar, but they're like little smaller. So they can be seagrass. So if you go to like any beach, you could probably sometimes see these big seaweeds and seagrass growing out there. So like the kelp forest, they can also trap the sunlight and carbon dioxide, and they can provide food to the other fishes. And there could be other algae, but I'm focused on microorganisms. So it's basically small microbes that you cannot see at all. And that also exists in the water, and they're commonly referred to as phytoplankton. So phytoplankton, meaning phyto is that they can like get their energy from the sunlight, and plankton is plant-like. So as I mentioned, primary producers drive all the life on Earth. So they can take up the light and carbon dioxide and produce organic carbon and oxygen. And we use that oxygen to breathe and survive. And the nice, interesting thing is that the microorganisms like cyanobacteria also can do the same thing. So they can easily grow anywhere in fresh or marine water. And they have chloroplasts in them that can trap this, similar to what plants have. So all of this is very important. So if you see a whole like globe, and if you spread it out on a map, and you can actually see how much phytoplankton is there in the water body. So if you see at this picture, all the light blue light is all the global phytoplankton present in our water bodies. And you can see that this is a lot of it. So all the ocean waters, all the lakes have a lot of cyanobacteria. So if you remove them, all the other fishes and plants will die in the ocean. So this is very important. And you can ask the question, why is this blue and green? So because they have pigments, so they're basically colored pigments. So similar, you have pigments in your eye that can trap the light and you can see everything in color. Similarly, cyanobacteria also have pigments. So they can absorb light within a specific wavelength, and when they emit light, they are like in the blue-green range. So that's why they are called cyanobacteria. Cyano means the color, which is like bluish-green color, and they are bacteria. Can you put up the second poll, Mrs. Bourne?
Sure. Let's see. Second poll would be environmental stressors.
Yes.
All right, here we go. Go ahead and answer the question, friends. What do you think environmental stressors include? The options are wind and light, temperature, nutrients, competition with predators, all of the above, or none of the above. So far, some people are answering in. Wait 1 more minute. All right, I'm going to go ahead and end the polling. It looks like most people thought all of the above.
That is perfect. Okay, everyone's smart here. That's good. So you're right. So when you see, like if I give you a tomato plant and I tell you, hey, I need this plant to grow, it needs to produce a lot of tomatoes. And you'd be like, okay, I can do this. But you yourself cannot do that. When you put them outside in your balcony on your garden, there are a lot of things that can like stop its growth. So it could be temperature, like if it's too cold, the plant doesn't survive. If it is too windy, the leaves might fall off, it won't survive. It needs enough light in the atmosphere to, for it to grow healthy. But interestingly, there are other things that also can stop it. So if you have a caterpillar infestation, it comes, eat away your tomatoes, you don't have any tomatoes. So similarly, even cyanobacteria are like exposed to a lot of stressors. So stressors meaning that something that can stop them from growing or something that can eat them up. So for example, cyanobacteria are affected by light and amount of UV radiation. Osmolarity, meaning the amount of salt in the water. If it's too salty, the cyanobacteria doesn't like to grow. It could be nutrients. So cyanobacteria love nutrients like nitrogen and phosphorus. And most importantly, it can also have an issue with bacteria. So it wants to compete with other bacteria for space or for energy and stuff like that. And the other thing is grazers. So that is what my research is focused on. So cyanobacteria, as I've shown in the first video, they can be eaten by a lot of other microorganisms. So for example, you can see here there are two grazers that I put up. One is an amoeba, like, the cartoon that had those 3 amoeba, similar to that. And the other one is a ciliate. And the ciliate is what I work on. So it is another microorganism, and it has like these small hairs which allow it to go from place to place. And it has actually a mouth, interestingly. And it goes and eats all the cyanobacteria in the water, which is very interesting. Mrs. Bond, can you put up the next poll?
Yes, I most definitely can. All right. The next poll is examples of a predator-prey relationship. All right, so the example of predator-prey relationship. The options are a human eating fruit, a lion eating a zebra, or a zebra eating grass. Go ahead and get your answers in. And if you have questions, you can go ahead and put them in the question answer slot. And there is one question that's coming while the polls are coming in.
Mm-hmm.
Does bacteria grow in the sea?
Can you repeat that, please?
Does bacteria grow in the sea?
Yes. Like the water contains millions and trillions of bacteria. So it's interesting because my lab actually goes, I'll show you a picture. We go out to the ocean and collect samples, and you can filter the water. And when you take the filter paper and put it under the microscope, you can see millions of microorganisms. And mostly they are dominated by cyanobacteria because they can grow easily with light and carbon dioxide, which is like available a lot in our atmosphere. So yes, there are a lot of bacteria. That's a good question.
That's a very good question. All right, we have the end to our polling. It looks like everyone thinks a predator-prey relationship is a lion eating a zebra.
That's right. So that's awesome. So the same thing. So when you see in microscopic level, the predator-prey relationship is also similar here. So the predator in our case is a ciliate or an amoeba, and those love to eat the cyanobacteria. So that's interesting that they like to eat and they get their energy from them. So okay, so till now I spoke about I'm working on like to understand what cyanobacteria is eating. But the question comes, why am I even doing this research? Why is people looking at it? So it's very important because we need to like kind of find an alternative to fossil fuels. By fossil fuel, I mean that, millions of years ago, there are a lot of organisms and like animals that died. And the most interesting thing is that once they die, once they get buried in the soil, they undergo this process where you can actually extract like gas from them. So we get all our gasoline that you can drive your cars with from fossil fuels. But unfortunately, like fossil fuels have a bigger issue. Mrs. Bond, can you put up the question, the poll?
Yes. Let's see, the next question is, are fossil fuels, are they renewable? True or false? An answer in that question. And remember, friends, if you have questions while you're hearing about what's going on in the lecture, you can also type them in and I will ask in the next break as well. So far, it looks like people think, I'm going to share the results, that fossil fuels, it's false.
So that's true. That's awesome. So renewable means that you can always, you have a lot of supply and they can grow again. So it's like renewable can be, if you have a fruit, you pick the fruit, the plant can produce more fruit. So, you can keep getting energy from it. But unfortunately, fossil fuels, once you dig up the earth and you take everything out, there's nothing that can replace it. So we need to see alternatives. So the nice thing is that scientists are very clever. And they've found that you could actually use a lot of like plants, so the inedible plants, the weeds, you could actually extract like lignin from them and you could use them to produce energy. But unfortunately, in like when you see crops, you have to cut down big trees that has a lot of issues with them. So scientists, they thought of another idea. They were like, oh, let's look at algae, because as I've mentioned, cyanobacteria and algae, they can grow pretty much anywhere. So you could use actually them to extract oils, and you can extract like gas from them, literally. So there are a lot of like smart chemical engineers who can use this algae and then extract them, which is like so nice. So this is example of a photosynthetic factory. Okay, so this is cyanobacteria in like big ponds. So you can grow them. So they expose to a lot of light and carbon dioxide, so they can grow really fast. So the nice thing is that they can be grown in large volumes and you can extract a lot of important products from them. But unfortunately, as I've mentioned, when you grow them outside, a lot of things like to eat them. And this causes something called a pond crash. So as you can see, they're supposed to be green, but because they get eaten away by the ciliates and amoeba, they become brown and you cannot use them anymore. So this is where my work comes into play. So what I'm trying to do is like actually look at the genome, like look at the genes of cyanobacteria and see, can I identify specific genes that can help them fight against these like predators. So if you imagine like the Harry Potter, imagine I make a cloak for the cyanobacteria, like an invisibility cloak, so that, the predators cannot see them, which will be very interesting. So what I do is I do a lot of genetic studies. So because genes are very important for function, so I'm trying to see if I identify a specific gene that can, help these cyanobacteria and protect them from being eaten by ciliates, which is pretty interesting. So this is what I work on. So you can see the green color is actually the color of the cyanobacteria. And by chromosome, I mean that's where all the genes are sitting, and genes are responsible for functions. So the last part of my research is that, okay, they can be used for industries and everything, but they actually can solve very important environmental problems also. So my research can be used to understand like how and why these harmful algal blooms are formed. So if people are from San Diego, you know that we always have a lot of dinoflagellate and algal blooms in the ocean. And this is partly because of us, we put a lot of nutrients in the soil, which goes into the oceans. And because of this, the cyanobacteria grow a lot. And once they grow a lot, they cover up all the lakes and the ocean. So this is, for example, in Ohio, Lake Erie. And this was in 2011, they had a bad explosive growth of cyanobacteria. And what happens is once they cover the layer of the lake, the light won't go inside. So the other marine organisms and the fishes cannot survive because they also require light. So this is like a bigger issue. So in this case, you can actually use these grazers like the ciliates and the predators to eat the cyanobacteria. So my work is basically studying like how genes can be used to study both harmful algal blooms and also how they could be used in industries.
Oh, great.
Yeah, that's it.
There was a really good question from Darcy. She
Yes.
So if there's a benefit to CO2 gas output from using microbacteria over using fossil fuels.
That's very true. So fossil fuels emit a lot of carbon dioxide. So they produce a lot of methane gas, which causes like, all these like global big effects and climate change and all those things. So you're right. So if we switch to biofuels, that could because cyanobacteria take up the carbon dioxide. So they can actually reduce the greenhouse gases and which could reduce global warming in big picture. That's a very good question.
That's a really good question. And there's another question. Are they in our pools at our house?
They're pretty much everywhere. Yeah, they're not in the amounts that are like toxic to you, but actually in your pools you put chlorine, right? Like you put the bleach, and that basically kills majority of the bacteria. But yeah, bacteria are actually very good for us. So yeah, they are in your pools if you don't clean them.
We've been hearing a lot about microscopic viruses that are around. Are all bacteria and viruses bad for us, or are some of them good?
No, actually, when you look at your own body, you actually have a lot of bacteria and they are in your gut. So actually, they are very important. There are a lot of viruses and bacteria that are very bad. So like COVID-19 or SARS-CoV-2 is very harmful because they cause a lot of these diseases. But there are a lot of bacteria which are also helpful. So for example, the bacteria in your stomach, it's called Clostridium. And it's very important because it protects you from other bacteria. So other bacteria that are harmful to you, these Clostridium can come and eat them away. So they can actually protect you from like big diseases, and they're important for your gut health. So you need to eat healthy to keep your gut system very strong.
And are biofuels other things like wood?
Yes. Yeah. So you could extract biofuel technically from wood. But the unfortunate thing is that in wood, there's a substance called lignin, and you cannot break it down. It's very difficult. Its molecular structure is very strong. So that's why researchers have moved from using plants and trees for biofuels to using microorganisms for biofuels.
Awesome. I would love to hear a little bit more about what your workday looks like when you go to work.
Yeah. So, I majority of time work in the lab, so I'm on my feet a lot of time. So I go to the lab, I set up experiments, which is like really cool. So yeah, so I set up a lot of experiments. So I keep walking around labs. So when I go to the lab, the first thing I do is wear my protective goggles and my lab coat, which is very important. Like if by mistake any chemical spills on me, I'm protecting myself and my clothes. I always wear gloves and then, shoes. Like people don't think shoes are important, but not only the shoes have to be comfortable, but they're supposed to be closed-toed. So you cannot show your little tippy toes, because if something falls on you and then it can like burn your feet. So, which is good. So that's number one. So your safety is number one. And then basically I do experiments. So my cyanobacteria take 5 days to grow. So I usually plan an experiment 5 days ahead of time. I let them grow. So these are how my typical experiments look like. I'll play this video. So I can grow cyanobacteria. The cool thing is that they can grow in flasks. They can grow in like test tubes, but they also can grow in these small micro-tighter dishes. So I can grow big volumes to small volumes. And so this is an example of a cyanobacteria culture that doesn't have any ciliate. So ciliate means like these things that are going around eating them. And I add different amounts of like predator to them to see how they die. So you can see that the minute I add any predator to them, they die and become like yellow in color. So apart from that, I also do microscopy. So I love taking pictures, which is very interesting. And the nice thing is that cyanobacteria, they are fluorescent. So when you see them under the microscope, they are like bright red in color, which is really pretty. And the other thing I do, because I do a lot of work with genes, I do sequencing. So basically, I study the DNA and how like the 4 nucleotides in the DNA are arranged. So I do a lot of sequencing as well. Yeah, so the next is, where is my lab located? I am super lucky. I'm right on the ocean. So this is Scripps Institute of Oceanography. It's a big institution. They not only do marine biology, but they do atmospheric sciences. They do physical oceanography. They are very cool. They collaborate with NASA. Actually, there are people who have sent cyanobacteria to space to see, how cyanobacteria react in space. And the reason they're doing that is that, once we take over Mars, which is our ultimate goal, to go to Mars and stay there, is that you need microorganisms to survive there. So actually, like, people who I work with are like trying to send samples to Mars, and which is very interesting. So this is where my institute is. My office actually has a view of the ocean. So every day I can see very beautiful sunsets, and sometimes I see lots of dolphins. The other day I saw a gray whale, which is really interesting. So from time to time we see a lot of whales, and you see humpback whale, it was very cute, and that was my first time seeing one. And so if you know La Jolla Shores, there's this big long pier, and the public cannot go there, but most scientists can go there. So the other thing we do is that we actually walk on this pier, go to the edge of the pier, drop a bucket, and take water. And we have been collecting water for 20 years. So the cool thing is that we can actually see how the water has changed over years. So, sometimes there are a lot of microorganisms in one season, and the other time the microorganism totally disappears. So this is very informative, because it helps you understand how ocean changes and how microorganisms change, how the predator-prey relationships takes place there. So we have a big repository of all these samples that we can go back and look, and we can store them in the fridge. So it's a very cold fridge, so it's -80. And you can store the samples, everything will be preserved, and you can take it out anytime, and you can look at different microorganisms under the scope, which is really interesting. Yeah, so, okay, the collaboration. So, we scientists cannot do things by ourselves. So we need help always. So we call it collaborations, or basically, help from other scientists. So I actually work with scientists at the University of California, the San Diego campus. And I work with 3 scientists. Susan Golden, who's very famous because she was the one who identified that microorganisms also have a clock, like how we have a biological clock inside us. So when it's at nighttime, the minute our body says, okay, it's time to shut off and go to sleep, at 6 or 7 in the morning, our body says it's time to wake up. So you have an internal clock. Even if you don't see a physical clock, we do that. And she was a scientist who was very famous because she identified that microorganisms also do the same thing. They also have a clock, which is pretty cool. So I work with a lot of scientists because I need their help. So I go to different labs to use different equipment. So I don't have a very cool microscope in my lab, but my neighboring lab has a beautiful microscope. So I go to them and be like, hey, can I use it? So I work with a lot of other scientists, and they help me, get my research going forward, which is cool.
That's great. I love collaborating with people. It's really fun.
Yeah.
Do when you're not at work?
Oh, okay. So when I'm not at work, usually I paint. I like doing a lot of art. So that's my go-to thing. Yeah, that's what I do. I actually dance. So, if my neighbors are not there, I usually put on music and dance. Yeah, those are the 2 things that I love doing.
Awesome. So it's not just science all the time. You do other.
No, and I actually volunteer at the Birch Aquarium. So if you have seen me at Birch Aquarium, next time come and say hi. I'm usually in the tide pool area, and I try to teach kids what are tide pools. And you should come visit the Birch Aquarium. It's really cool. They have really nice animals, and you can go touch them, which is super interesting. So I enjoy volunteering at the tide pools. It's fun.
Oh, that sounds awesome. What's the part of your job?
I didn't get you.
What's the hardest part of your job?
Hardest part of my job is like when an experiment fails. It's disappointing because you put in so much work and you think about a lot of things. You're like, okay, I should do this, this, and this, and everything doesn't go to plan. Like on a piece of paper, you can write down and say that I need, this result to come. But most of the time, like 99% of the time, you're getting something totally different. So you have to go back and see what was different, why was it happening, and sometimes it's very disappointing. So you have to keep motivating yourself to go back and repeat it and see if you're getting something different. And which is what like most scientists are used to. Like as time goes, we get used to defeat or like having negative results. But at the end, we make it work. Yeah.
That's great. Yeah, it's kind of like what we're experiencing now. The whole world is learning that scientists have things that they don't know yet. We're just all figuring it out. All right. What was it? What brought you to science? How did you become interested in it?
Yeah. So it is weird because when I was growing up, I never liked school. Like I used to go to school, but to go to do sports. So I was a very good athlete when I was growing up. So I used to go to school actually an hour before. So I used to go when the school started at 9. I used to go at 7:30, not to study, but to go run on the track. And I always used to get punished because my school clothes used to get dirty. So it was, and that's an interesting thing because I want to say, so I am not from the US, I am from India. And in India, the teachers are strict, like you cannot disobey them. And all schools in India have a school uniform. So you cannot wear your normal clothes and go. So we had a school uniform, you had to like cut your nails, no hair color, you got to like braid your hair. It was very strict. And I used to always get in trouble for having like spoiled my clothes before I started school, which was unfortunate, but my parents got used to it really quick. So every time they were called into, they were like, it's fine, whatever. But I think in 8th grade is when I realized that I wanted to become a scientist because all my friends wanted to become doctors and I didn't want to become a doctor. So I was talking to my mom and she's like, oh, why don't you become a scientist? Like, you need scientists for medicine to know all the things, which is when I thought that I was going to do my PhD. And that's when I learned about a PhD. And I was like, okay, I'm going to do a PhD. So I was in India. So I did my schooling in India. I got my bachelor's in microbiology in India and my master's in India. And after I did my master's, I was like, okay, I need to take a break. So I actually went to industry. So I basically went to a biotech company. And the nice thing is that they worked on vaccines. So I got to learn how to handle mice and guinea pigs and like, do injections for them. Because unfortunately, how much ever I love animals, you need to test it on them to see if it's not harmful to us. So, so I got trained in that, which was very interesting. And then I decided that, okay, I really enjoy doing science. I like going to lab and doing experiments. So I decided that I was going to apply for my PhD and get more training. So I applied to the US because I was like, it'll be a nice experience for me to leave my country and go to a new country independently and like, explore it, which is nice. So I actually came to T. So I got a degree from the University of Tennessee. And the part I loved about the program was that even though it was a university, I could go to the national lab, which was very cool. So this is Oak Ridge National Lab, as you can see here. And the cool thing about this lab is that they actually made the bombs that were dropped on Hiroshima and Nagasaki. So it was very interesting and they have very cool instruments. So that's where I did my PhD. And then after that, I wanted to get more training. So I moved to California. So I moved to San Diego. And that's where I am. So it's been a year in San Diego and it's been a fun experience.
That's great. Was Tennessee a lot different than California for people who have never been to Tennessee before?
Oh, it's a lot different. It's green. They have a lot of mountains, and yeah, it's like a very small town. So it's not a big city like San Diego is. So it was very different. But I think it was a good transition for me from coming from India to Tennessee. And the national park, Great Smoky Mountains, is there, which I love going. So we used to do a lot of hiking, which was nice, and it was nice. I mean, we had seasons there, which I really miss in San Diego.
Yeah, we don't have many seasons here.
No.
Role model or hero that you had when you were going to school, thinking about studying?
I think it would be my mom, and then my biology teacher. My mom never pressured me to study. She was always like, do what you want to do, which made me like get more interested in education and in science. When I told her I wanted to become an athlete, she was like, sure, if that's what you want to do, you got to like chase your dream. So and then I realized that, oh, I am pretty good at studying, so why don't I explore this? So, which is cool. And then I think my biology teacher in my 9th grade, she made science very interesting. So, I mean, she was awesome. She used to get all these props and explain like concepts at a basic level. Yeah, so that's what made me interested in science.
And did you have any obstacles that you had to overcome when you were becoming a scientist?
It was a lot. It was difficult. Once I moved from India to US, it was very difficult because like labs in India are run differently. Well, like for example, I didn't know how to use like, how to like light a, like it's called a burner. So I didn't know how to use things here because everything was so different. So the day one when I came here, when I went to lab, I had to ask so many people for help because I didn't know how things worked here. So it was very different. And then in India, everything is the opposite. So you drive on the other side of the road. So that was an adjustment to take. And then the metric system, everything is different here. So I had to like relearn all my metric numbers and everything. And so it was challenging, but I think I had awesome people around me who took the time to explain things. So that's why you need to be nice to people. Is like, when you go out of country, you get a lot of help. So yeah, it was different, but I think now I'm getting a hold of how things run here.
That's awesome. Have things changed with your work life with COVID-19?
Oh, well, yeah, actually, because, most scientists, like not all, most scientists actually have to go to lab and do experiments, which was difficult once our lab closed because we had to do social distancing. So I had to like put all my cultures in the fridge and store them, which was sad. And I actually planned a lot of experiments and I had to put everything on hold. Also, like because I do a lot of experiments, I don't have any work from home. But fortunately for me, because I also work with DNA and I do sequencing, I'm like switching to like sitting on the computer and doing a lot of data analysis and reading a lot, which has been interesting. I mean, I read a lot of papers, like most scientists have to keep up to date on what's been published. So I basically sit and read a few papers every day. And even though it had been challenging, I think it was a welcome surprise because now it has made me appreciate the other things that I need to like focus on and, which is going to help me in my research. So it's been fun. Yeah, I can't wait to go back to lab and do experiments.
I'm sure our students kind of feel the same way. I'm sure.
Yeah.
School.
Yeah, so I think like, for your kid, for now, when you guys go back to like school and, you have online classes, it'll be an interesting challenge both for the teacher and the students. But I think it'll be interesting, like, you don't have to get up and get ready to school, which sounds fun, which is good.
Yes, there's good things too.
Yeah.
All right, there was one question that we didn't get to, and it was about why are the bacteria green?
Oh, the bacteria are green because of the pigment. So they have the pigment called chlorophyll. So that's why your trees are green, because of the pigment chlorophyll. And the nice thing is that these pigments can absorb light. So in light, there are different colors. Like, when you see light, it might look colorless or white to you, but actually when you split the light, there are different colors. So the Vijaya, the violet, indigo, the red, orange, green, all those colors, you can actually split them. And the cyanobacteria can actually absorb in the red range, and it can emit in the blue and green range. So basically, the pigments give color. It's actually why all of us have different color of our eyes. So I have brown eyes, but then other people can have blue. So depending on how much pigment you produce, you can have different colored eyes too.
That's great. Super interesting. I saw somebody raise their hand. If you have a question, if you type it in the question box, then I'll be able to see what your question is. But I wanted to just ask maybe another question about when you were studying and you were at the different universities and you had all those people that were helping you. Is that how scientists usually are? Are they usually helpful to each other and sharing information? How does it work when you have a new article or journal article that comes out?
Yeah, so yeah, all scientists do collaborations. So they all work together. And the nice thing is that, when you do science, it's not cheap to do science. You need these big equipments. You need to buy a lot of reagents. So everything that you see here, you need a lot of money to buy them. But the easier way to do is to share. So if I don't have a microscope, but then my lab beside me has a microscope, we always share resources, which is useful because then I don't need to spend millions of dollars and buy an equipment that, I might just use for like one day or like few months, and then I don't have any use for it. So in science, collaborations are very important. Also, when you do research, if by myself, if I have to do this research, it'll take me 10 years at least to complete this research. But if I have help around me, if I have other scientists who are going to come and ask me, oh, can I help you with this? Or if I go and ask them hey, can you do this experiment for me? You're an expert in this. Why don't you finish this part for me? And that's how you get work done really fast. So even for producing a vaccine, if one scientist has to work on a vaccine, it's impossible to find a vaccine. So you need a team of scientists to come together. And because all of our specialties are so different, like I might be good in microbiology, but there may be other people who are good with viruses, and they can give their input on how your research can be improved. So that's how we do. We also do a lot of lab meetings. So there'll be a few labs who come together and we discuss our work. If you have any issues in science, you can talk to them and you can ask them questions, and if they know a technique, they can help you with the questions. So that's how you also improve your thinking. You can give input to people in other fields even though you're not an expert. But I think collaborations are very important and they're key. So it's cool because you can get research done really fast and you can actually know more people, and in a bigger picture, you can do networking, you can talk to them, you can improve your communication skills. So all of these are very important.
That's awesome. Yeah, I love that part of science too.
Yeah.
One more question relating to your research. It was how much algae would you need to power a specific thing, like a house or a light, or how much is needed?
Well, you need a lot of them. So if you see this slide, so this is all these are huge tanks of water with gallons and gallons of water. So you'll need a lot of them. But the nice thing about them is that because they grow so fast, you can grow a lot of them really fast. So you don't have to worry about it. And the interesting thing is that now they're actually trying to use cyanobacteria in solar panels. So they are trying to grow these cyanobacteria in the solar panels. So they can just put it above your house, and that can directly trap the energy and light, and you could use it to run all the equipments. So yeah, you need a lot, but then because they're easy to grow, you could grow a lot of them.
That's wonderful. We have a lot of questions for us, and We really appreciate you taking the time to share about your research and about your process of becoming a scientist. You're very inspirational. Thank you very much, Dr. Kumar.
Oh, thank you so much for having me.
And we have another webinar next week, friends. I can't release yet who the panelist is, but I have 2 really special people lined up, and I will be emailing you on Monday with the login and telling you what our topic will be for next week. And I'm looking forward to seeing you then too. Again, thank you so much, Dr. Kumar. We really appreciate your time and your expertise. And friends, make sure to stay kind, stay curious, and wash your hands. Thank you, everyone.
Bye.